Hi students, my name is Jatin Dewan and today we shall be studying about strategies for enhancement in food production. The first one is animal husbandry. So what is animal husbandry? It mainly deals with the care and breeding of livestock that are very much useful to humans. This can be achieved through new technologies for better quality and quantity. It is agricultural practice of breeding and raising livestock. It also includes farming and fisheries. Fisheries would include rearing, catching, selling of and other things of fish, molluscs like shellfish, and crustaceans like prawns and crabs, etc. Then the next one would be poultry farm management. Poultry is the class of domesticated fowl birds used for food for their eggs and meat. It includes chicken, ducks, turkey, and geese. These are the certain points you gotta keep in mind. Proper food and water is required, safe farm management, hygiene and healthcare, selection of disease free and suitable breeds. The next topic is dairy farm management. Dairying is the management of animals for milk and its products for human consumption. In this, people mainly deal with the processes and systems that increase yield and improve the quality of milk. These are several points which are better for maintaining the regularity of dairy farm. Proper shelter should be provided, sufficient water should be available, selection of good breeds having high yield potential, good quality and quantity of fodder should be provided, cleanliness and hygiene should be maintained at all times. The next topic is beekeeping and which is also known as apiculture. It is the maintenance of hives of honeybees or for the production of honey. Beekeeping can be practiced in any area where there are sufficient bee pastures of some wild shrubs, fruit orchards, and cultivated crops. Several points for maintaining a healthy apiculture would be selection of suitable location for keeping the beehives, maintaining and management of beehives during different seasons, handling and collection of honey and bee wax, proper knowledge of nature and habits of bee, catching and hiving of swarms should be done with full protection and full precautions. The next topic is breeding. Breeding of animals is a very important aspect in animal husbandry. The main aim is to increase the quality of yield and improving the desirable qualities of produce. Breeding is two types: inbreeding and outbreeding. In inbreeding, it refers to the mating of more closely related individuals within the same breed of 4 to 6 generations. In this, superior males and females are identified and are made to mate. The progeny thus obtained are evaluated and are identified for further mating. Outbreeding is defined as the breeding of unrelated animals, either of the same breed or of different breed or different species but having common ancestors for 4 to 6 generations. Then outcrossing. Outcrossing would mean that there is mating of animals within the same breed but having no common ancestor on either side of the pedigree up to 4 to 6 generations. In crossbreeding, superior males of one breed are mated with superior female of another breed but of same species. It also allows the desirable qualities of two different breeds to be combined together. Then there is interspecific hybridization. Male and female of two different species are made to cross combine desirable characters. There are several techniques through which these things are done. One is artificial insemination. In artificial insemination, semen from desired animal is stored and in frozen form because it becomes easier to transport so that it can be used whenever required. The second one is multiple ovulation embryo transfer. In this, cow is administered to induce ovulation, producing 6 to 8 eggs after that mating is done, either directly or through artificial insemination. Then fertilized eggs at 8 to 32 blastomere stage were recovered and were finally transferred to the surrogate mothers. The next topic will be plant breeding. In plant breeding, the process is initiated by getting the proper yield, having better quality which is resistant to pathogens and has increased tolerance to environmental stress. The steps involved are germplasm collection, evaluation and selection of parents, and then crosses are made, selection and testing of superior recombinants. The further development for disease resistance. Resistance of the host plant is ability to prevent the pathogens from causing disease and is determined by the genetic constitution of the host plant. Methods are as follows. Conventional method will be the first one. It is the hybridization and selection technique in which we screen the germplasm for having the resistant sources. Then hybridization is done for the selected plants, and then later on selection and evaluation of hybrids is done and tested and released for new varieties. The next one is mutation. Mutation is a process by which genetic variations are created with various changes in base sequences with genes, resulting in creation of a new character or trait which is not found in parental type. It can also be done through radiations on chemicals. The next one is biofortification. Biofortification is the breeding of crops with higher levels of vitamins, minerals, or higher protein and healthier fats. It is most practical means to improve public health. It can improve protein content and quality, oil content and quality, vitamin content, micronutrient and mineral content. The next is SCP or single cell protein. In single cell protein, the microbes are grown on a very large scale and are used as nutrient rich food. Example in its spirulina. Spirulina, then there would be several uses of single cell protein. Single cell protein are rich in proteins and low in fats. They can easily be grown on cheaper materials like wastewater, etc. They act as a supply of fertilizer and can reduce the water pollution. The next topic is tissue culture. It is a technique in which a whole plant is regenerated from any part of plant by growing it under suitable conditions and in suitable media. Like the medium has to be chosen correctly. Uses. Now, a large number of plants can be grown in very short period of time. Healthy and disease-free plant can be grown by meristem culture. The last topic we'll have for this chapter is somatic hybridization. Somatic hybridization is defined as a process of fusion of protoplast of somatic cells of two different varieties or species. The steps involved are: first, the isolation of protoplast is done, then the fusion is considered, and then later on growth of fused protoplast to form somatic hybrid. Example in tomato is waited and considered to get over so that we can get a new hybrid. I hope you understood this lesson. Several abbreviations I would like to dictate which are very important for this chapter are ET, which would stand for embryo transfer, IARI, which is the Indian Agriculture Research Institute, IRRI, which is International Rice Research Institute, then NDRI, which is the National Dairy Research Institute, MOET, multiple ovulation embryo transfer, SCP, single cell protein, ICAR, Indian Council of Agriculture Research. If you understood the lesson well, please follow me and don't forget to comment below.